Welcome to this summary video of National 5 Biology, Unit 2, Multicellular Organisms. We're going to be looking at producing new cells today. We'll start by looking at chromosomes, and you would find chromosomes if you magnified the nucleus of a cell where genetic information is found. Chromosomes themselves are made up of chromatids, and they are joined in the middle by a structure called the centromere. A section of a chromosome is called the gene, which is DNA which codes for a protein, which we'll look at in a later key area. In terms of the number of chromosomes in living organisms, every species of living organism has a characteristic number of chromosomes, and we call this its chromosome complement. So for example, humans have 46 chromosomes, or 23 pairs, dogs have 78, potato plants have 24. Every species of living organism has their own characteristic number of chromosomes, and we call this a chromosome complement. In terms of the number of chromosomes in cells, a cell that contains a single set of chromosomes is called haploid, whereas a cell that contains two matching sets of chromosomes are called diploid, di meaning two. All normal body cells, for example skin cells, muscle cells, liver cells, are all diploid. That's the standard number of chromosomes we find in our normal body cells. However, sex cells, or gametes, such as sperm and egg, are haploid. They only contain a single set of chromosomes. And that's because a haploid egg and a haploid sperm are going to combine during fertilization to produce a diploid zygote. But we'll come to this in a different key area. The way that we make new cells, or cell division, is a process called mitosis, and it is required to produce new cells for both growth and for the repair of damaged cells. And this is why we need mitosis. The other thing that happens during this is while new cells are produced from an original cell, which we call the mother cell, they produce two genetically identical daughter cells, and these maintain the diploid chromosome complement. What this means is that the number of chromosomes in the mother cell will be the same as the number of chromosomes in each daughter cell. They are not halved each time. They maintain that same number of chromosomes, so they maintain the diploid chromosome complement. We're now going to take a quick look at the stages of mitosis, so how cell division takes place. At the start, chromosomes become more visible as they double up to form two identical chromatids. So these become visible when cell division is about to take place. The second stage, the chromosomes will shorten and thicken, and we can now see this distinctive chromosome shape. The chromosome is made up of the two chromatids, which are joined in the middle by a centromere. In stage 3, the chromosomes line up at the equator, and the membrane surrounding the nucleus disintegrates, and a structure called spindle fibres starts to fill the cell. These spindle fibres then attach to the centromere of each chromosome, which is lined up at the equator. In the next stage, spindle fibres pull the chromatids apart, and they pull these chromatids to the opposite ends, or what we would call poles of the cell. So if you imagine the cell as a globe, the chromosomes line up at the equator, and the chromatids are pulled to the opposite poles of the cell. Finally, a nuclear membrane will now form around each set of chromosomes at the top and at the bottom of the cell at the opposite poles. The cell then pinches in the middle, and it's divided into those two genetically identical daughter cells. So for one mother cell, we now have two genetically identical daughter cells, which has maintained the chromosome complement. In terms of different types of cells, we have specialised cells, which are able to perform a certain function. So for example, skin cells, sperm cells, brain cells. These have one job, and they can only replicate to become that same form of cell. So skin cells can only replicate to form new skin cells, for example. They cannot become a different type of cell. And this is what we call specialised. However, there are unspecialised forms of cells, and we call these stem cells in humans and animals. Stem cells are unspecialised cells that are able to divide to produce more stem cells, a process that we say is self-renewing. So a single stem cell can make more stem cells, or they can produce cells that then go on to become specialised cells. So a stem cell could produce a stem cell that then differentiates into a skin cell, or a red blood cell, or a muscle cell, for example. And this is why these are very important in medicine and research. Finally, there's something that we call a hierarchy of cells. And our cells can be specialised in order to work together to form tissues, again, like skin or muscle. These tissues can then form organs, and these groups of organs can work together in order to form systems. So it's important you know the hierarchy, the ranking of these are cells, tissues, organs, and then systems.